Good afternoon, everyone. My name is Chandrasimha. I'm an undergraduate student at the University of Arkansas at Little Rock, and I'd like to discuss with you today about my research project studying the antibacterial efficacy of nanostructures on nickel titanium archwires for orthodontic applications. And this is the outline of my presentation that I'm going to be giving today. To give you a little background of my research, there are currently over 4 million patients undergoing orthodontic treatment in the United States alone. An orthodontic treatment puts patients at a greater risk of developing iatrogenic diseases because the orthodontic devices themselves are a scaffold for dental plaque accumulation. And this dental plaque induces the formation of caries, gingivitis, and periodontitis. And this brings us to the purpose of my experiment, which was to devise a novel, non-invasive, low-cost method to curtail periodontitis diseases through bacterial inhibition method. We aim to inhibit bacterial growth in the oral cavity by engineering nanostructures on orthodontic archwires. Nanostructures exhibit antibacterial properties, and they are a non-invasive procedure because they can be manufactured onto orthodontic devices before being applied into patients. And their synthesis method is a low-cost method using hot water treatment, which the schematic is outlined in figure 2 to the right. And why we chose Staphylococcus epidermidis as our bacterial to study was because of its implications in dental diseases and its pathogenic synergism in the oral cavity. To induce these nanostructures using hot water treatment method, we first clean the wires using sulfuric acid, and then they were rinsed in DI water, followed by treatment in hot water at 95 degrees Celsius for 10 hours. Figure 3 depicts the mechanism of formation of these metal oxide nanostructures, or monsters for short. To characterize the antibacterial efficacy of these nanostructures on these archwires, the archwires were placed onto bacterial lawns, which were spread with Staphylococcus epidermidis, and they were incubated at 37 degrees Celsius under airway conditions for 1 day, and the images obtained were used using gel. Following a modified kirby-bauver test, these wires were removed and washed, and then sonicated in PBS to remove any bacteria that were adhering onto the wires. These bacteria were then diluted and plated onto lawns to count the number of colonies that formed from the bacteria. This provides us a method to quantify the number of bacteria on the wires, and it allows us to analyze the antibacterial efficacy of these wires. To better represent the oral microenvironment, which has periods of static and dynamic motion, we conducted an additional antibacterial test by incubating the wires on a bacterial broth solution on a shaker. By doing so, we hope to better represent the dynamic nature of the oral cavity, which has periods of fluidic motion when you consume fluids in your mouth. And on the figure to the right, we have a depiction of the bacterial adhesion onto the archwires. In this slide, you can see the SEM images of the treated and untreated nickel titanium archwires. To the left, you have the untreated nickel titanium archwires without nanostructures, and you can see how the surface is really smooth. And to the right, you have the nanostructured nickel titanium archwires, which are a lot rougher and thus causing mechanical lysis of bacteria. And this slide shows the images of the archwires placed onto the bacterial lawns in the experiment. This table depicts the colony counting method for the bacterial lawn and the results that it produced. As you can see visually, positive control has more bacterial colonies on average than a nanostructured wire. And this tells us that the nanostructured wire is better at inhibiting bacterial growth. Table 3 depicts the results of the colony counting method of the bacterial broth on the shaker. As you can see, the positive control in the nanostructured wire did not have any bacteria colonies on them, indicating that a dynamic oral cavity prevents bacteria from adhering to orthodontic devices. Statistical analysis was conducted on the MKB test to determine if the colony-forming units were statistically significant from the positive control versus the nanostructured wire. And by doing so, we can determine if the nanostructures are in fact exhibiting antibacterial properties. And if you look at the data here, we see that the t-test for unequal variance produced a p-value less than 0.05. And when the p-value is less than 0.05, we know that our data is statistically significant. And that indicates that the archwires and the nanostructures are indeed exhibiting antibacterial properties. Given the results of my experiment, we can infer that a dynamic fluid environment decreases the incidence of biofilm adhesion on archwires. This is supported from our research findings of the bacterial broth on a shaker, which had no bacterial growth on either of the controls, on either of the groups. However, in contrast, the static environment of the bacterial lawn did have bacterial colonies that grew on them, and we found a statistical significance between the nanostructured group, which had monsters, and the positive control group, indicating that there are antibacterial properties on these monsters. And previous studies have found a positive correlation between surface roughness and biofilm adhesion. So as surface roughness increases, biofilm adhesion should increase. However, in our study, we found that while nanostructures should increase surface roughness and biofilm adhesion, which means that there should be more bacteria on nanostructures, we found that there was a decrease in bacterial growth on nanostructured group. So we hypothesized that nanostructures not only increase the biofilm adhesion, but they also kill more bacteria. So they have dual roles. For future research, we hope to study other metal alloys for their antibacterial properties and to characterize these nanomechanical properties to determine the structural features that best optimize nanostructures for antibacterial efficacy. And we hope to also conduct SEM studies to observe the biofilm at the micro level and to also determine the antibacterial mechanism of action of these nanostructures. We also hope in the future to do clinical trials to observe how these nanostructures function in the oral cavity. I would like to acknowledge the Signature Experience Grant for providing funding for this research, and to acknowledge Dr. Karabakak's lab and Dr. Bush's lab for the support and mentorship in this research project. I would also like to thank the Center for Integrative Nanotechnology Sciences at UALR and the help of Dr. Watnova and Hannah Hester for SEM imaging. And these are my study references. Thank you for listening to my presentation. If you have any questions, comments, or suggestions, please do let me know.